Hello, everybody. Welcome back. Let's go through just the very basics of neuroscience embryology. So, remember there's the central nervous system and the peripheral nervous system, and we can organize it into the segmental. Segmental is basically the CNS, and that's where the spinal cord and the brain stems are directly connected to the peripheral structures. The suprasegmental would be the cerebral hemisphere, the diencephalon, and the cerebellum. Those do not directly connect to the peripheral structures. Instead, it ends in the central nervous system. So it's really just limited to the brain and brainstem. Gray matters are going to be kind of different as you go down the spinal cord. It's going to be large in the cervical and the lumbar region because that's where the brachial and the lumbar plexus are, and that has a lot of fine motor neurons, of course. The thoracic region is the only one with the lateral horns right here. Well, this is actually cervical. Sorry, thoracic would be like right here, right here, right here, right here. Like it's very prominent, and the white matter is going to be most prominent in the cervical region, of course. So as the tracks like go down, the white matter decreases. And the ascending tracks will also end in the cervical segment. That's why there's so much of the white matter. So there's the third lateral ventricle, or the choroid plexus, and the fourth ventricle will merge right into the straight sinus. Straight sinus goes down and carries the CSF to the spinal canal. There's a narrow cavity in the midbrain, and that causes the aqueduct. That creates the aqueduct of Sylvius. So this is the general path of the CNS. So it comes down, goes through the straight sinus, goes up through here, goes around, gets distributed, or it goes from the eye or area above, gets distributed. The cortex is going to be the cerebral and cerebellar regions of the brain. Gray matter is made out of the neuronal cell bodies, along with the nerve fibers, some cells that supplement or like they maintain the fibers, and blood vessels. White matter is going to be your axons. Brainstem is going to be the gray matter included with the nuclei, and the columns would be the spinal cord gray matter. Nuclei is the collection of nerve bodies with the same function in the central nervous system, while the ganglia are the collection of nerve cell bodies with the same function in the peripheral nervous system. So here we go. There would be the dorsal and then the ventral root, ganglion right here, white matter, gray matter inside. There are neuropils, which are glial processes. That also includes the blood capillaries, axons, dendrites, synapses, and those form a network where the neuronal cell bodies are going to be embedded, and that predominates in the gray matter, and it's usually unmyelinated. The reticular formation is a type of neuron in the brainstem that is required for like wakefulness and alertness. Funiculi are just large bundles of tracts, and the tracts are the smaller bundles, and that conveys a specific type of information. So the tracts are like separated in the spinal cord, and then the reticular formation kind of like it's just what everything's embedded in. The commissure is the group of nerve fibers connecting similar regions, and there are like all these 5: the frontal to parietal, the parietal to occipital, and then the main one is the corpus callosum, which like link two hemispheres together. The decussation is going to be the crossing at the midline, and the main one is going to be the optic chiasm. The lemniscus is the main sensory pathway of the central nervous system, and it goes all the way up to the brain. And the pathway is a series of neuron to neuron links in the functional system. And the very first cell is going to be the receptor. Okay, so let's go through this really fast. Remember, there's going to be the nodal cord, and that gives rise to the neuroplate. And then the neuroectoderm will make the neurotube, the neurocrest cells, and the ectodermal placodes. And neurotube is the precursor to the central nervous system. It closes in 18 days, but the cranial and caudal ends are open as the neuropores. The neurocrest cells will eventually make the peripheral nervous system. The ectodermal placodes are going to be the precursors to your central or cranial sensory ganglia, the hypophysis, and the inner ear. The anterior pore closes on the 25th day at the 18th to 20th somite stage, and it makes the lateral terminalis. The lateral terminalis kind of go through here. And the posterior neuropore will close at the end at the 25th somite stage on the day 28. So, what's important to take away from here? Neurotube closes in 18 days, anterior neuropore closes on the 25th day, and the posterior neuropore closes at day 28. So the neuroectodermal tissue is going to thicken to the neuroplate, and the neuroplate border will separate the ectoderm from the neuroplate. It bends and eventually joins. That makes the crest, and that's when these cells will break away, making the neurocrest cell. That's where the terminology comes from. Eventually, it closes and disconnects, and that's why the neurocrest cells form the epidermis, right? So that's why this comes from the endoderm, or the ectoderm comes from the neurocrest cells. And then the nodal core will eventually degenerate and only persist as the nucleus pulposus of the intervertebral disc. And then the mesoderm cells will differentiate into the somites and whatnot. Okay, so like I said, the neurofolds fold together and fuse, and then the crest cells break away. The ectodermal placodes are when the neuroepithelial cells become incorporated into certain areas of the surface ectoderm. So they kind of like migrate away to whatever signals them, gets embedded and stays there. So in the cranial region, the caudal forebrain, or it comes from caudal forebrain to the rhombomer number 6. Forebrain is going to form the cornea and the head mesoderm. The hindbrain, or the pons, the medulla oblongata, the diencephalon, will also form the bones of the face, the middle ear, thyroid, teeth, cranial nerves 7, 9, and 10, along with the parasympathetic ganglia and cranial nerve sympathetic ganglia. So hindbrain creates quite a bit. The vagal area is going to be from somites 1 to 7, and that will eventually create the cardiac neurocrest cells, the enteric nervous system, and the parafollicular cells of the thymus. Sorry, here we go. So in the trunk, it's going to be from somite 8 to 28. The peripheral dorsal root ganglion, sympathetic and parasympathetic autonomic peripheral motor neurons will form from that. And then finally, in the lumbosacral region, it's going to be from somite 28 and beyond, and that is responsible for forming the enteric neurons. So the tube will close on day 25, or I'm sorry, day 18. I'm so sorry. And then there, the neuroepithelial cells will undergo mitosis, and that makes the layers. So it closes, makes the layers. And then by the 5th week, the matrix zone is going to divide, making the neuroblasts, which will form the neurons, and the spongioblasts, which make the neuroglias. And the mantle layer will make the gray matters as the fibers will move out. And then the neuroblasts will thicken the mantle into the alar, which is going to be the sensory area, and the basal, which is the motor plate. And together that will make the sulcus limitans. And then the neurons will accumulate between the intermediate horns and the sympathetic portions of the anterior nervous system on the or autonomic nervous system on the T1 to T12 levels. The two afferent columns of the alar lamina, so alar is sensory, will get the somatic efferent and visceral column axons from the dorsal root ganglia. And the efferent columns will give rise to the general visceral efferent column and the general somatic efferent column. So it's going to have connections to the intestines and the muscle. The marginal zone fibers will extend and make the white matter. Ependymal layer will close the spinal cord cavity and it lines the mantle layer. And in the general visceral efferent column, it's going to only be in the thoracolumbar and the sacral regions only. And in the general somatic efferent columns, it's going to persist throughout the entire spinal cords, and those innervate the muscle. So here is the sulcus limitans. Here is going to be the matrix zone, and then the mantle zone, and then the marginal zone. So marginal zones would be the furthest, matrix zones would be the innermost. The marginal zone is where the fibers will extend. Ependymal layer is going to just be on the innermost layer, and it closes the central canal eventually. So the marginal zone is where the neuroblast from the mantle nuclei will accumulate. So the mantle zone is really where the fibers are. Matrix is where the cell bodies are. And then the marginal zone is mostly made out of the white matter because that's where the fibers are going. The brain stem will also develop in the same matter as the spinal cord. And they all kind of develop along the vertebra in the first 2 months. But then the vertebral column grows faster than the spinal cord, and that's why the spinal cord levels differs in adults and in children. So the spinal cord level is about L3 for babies and L1 for adults. Why is that important? Because if you ever need like epidural or a spinal tap, you need to be aware of where the spinal cord is. You can't tap into that, you'll cause damage. You have to go below. So the meninges will form from the neurocrest cells and the mesenchyme around days 20 to 35. So it's all forming while the brain is forming. It's quite a quick and complex process, really. The leptomeninges are the arachnoid and pia matter, and the external layer will thicken into the dura matter. The CSF will form during the 5th week, and that will fill the subarachnoid space. The neuroepithelium will differentiate into the neuroblast and the glioblast. The glioblast will migrate from the ventricular zone into the intermediate and the marginal zones to make astrocytes or oligodendrocytes or ependymal cells. The microglia will come from the mesenchymal cells, and that comes from the bone marrow. So when the neuroepithelial cells stop making the progenitor, they will differentiate into the ependymal cells. So as you can see here is the roof plate, alar plate, sensory, basal plate, motor, floor plate, all of this ependymal. Inside, sulcus limitans, the dorsal root ganglion. Here is the arachnoid. Pia, you can hardly ever see, but you can always see the dura. Okay, so how does the brain develop? At the end of the 4th week, the cephalic part of the spinal cord will make the primary brain vesicles, and that is going to be the prosencephalon, mesencephalon, and rhobencephalon. In the 5th week, the prosencephalon and the rhobencephalon will subdivide to make the secondary vesicles. The telencephalon is going to be made out of the cerebral hemisphere and the lateral ventricles and the superior part of the 3rd ventricle. The diencephalon is going to be between the hemispheres. That's going to be most of the 3rd ventricle. The mesencephalon is going to be the midbrain. That is going to develop into the cerebral aqueduct. The metacephalon is going to develop into the pons and cerebellum along with the 4th aqueduct. And then the myelincephalon is going to make the medulla and the central canal. So you can see here. So first is the prosencephalon, mesencephalon, romencephalon. Prosencephalon will make the telencephalon, diencephalon. Mesencephalon and mesencephalon. And the romencephalon will make the metacephalon and the myelincephalon. And as you can see here, metacephalon will make the pons and cerebellum. And diencephalon will make all of these structures. Okay, so how does the brain form the way it is? There is going to be something called a pontine flexure right here in the middle of the romencephalon. And then the cervical flexure right at the junction of the romencephalon and the spinal cord. And then there's the cephalic, right here. Cephalic flexure at the midbrain. And then, all in all, it just makes this S shape, you know? Cephalic and cervical is going to be concave ventrally, while the pontine flexure is the only one that's convex. Alright, so in adults, the telencephalon will lengthen, and in the midbrain, it forms the fifth, third, fourth cranial nerves, along with the pineal gland. The tectum is going to be above the superior. We'll cover that when we go into the medulla. And then pons will be responsible for like cranial nerves 8, 7, 9. That's from the lateral to the medial lateral to medial organization. And then cranial nerve 12 lies between the pyramid and the olive. Okay, so formation of the medulla forms from the myelencephalon. The roof plate will widen, and then the alar plate and the basal lamina lengthens and are pushed back anteriorly, while the sulcus limitans divides. So opens, these are pushed. Nuclei will be submerged into the marginal layer. Above, so it's going to come from the matrix, right? And then it gets pushed down and kind of out, and then an extension goes like anteriorly, and that will make the olive. Ependymal cells will surround the medulla oblongata, and the choroid plexus will push into the roof plate, separating it and creating the open and the closed part of the medulla. The basal plate will give rise to the pyramids. We'll go through the structures and the functions of those later. But in the meantime, the closed part of the medulla has the central canal, has the sensory or the alar plate, which is the sensory nuclei of the cranials 5, 8, 9, 10, along with the gracial and the cuneate nuclei. And the basal plate is going to be the motor nuclei of cranial nerves 9, 10, and 11, or 9, 10, 11, and 12. And the floor plate is going to be made out of the pyramids, which are the corticospinal tracts. So up here would be the open, down here would be the closed. And then in the 4th, 5th month, the roof plate is going to be reabsorbed, and it makes the foramina of Luschka and Magendie. And the open part of the medulla expands into the cavity of the 4th ventricle. So here are your olives that are formed. Here's the sulcus. We'll get all through that later. But more importantly, right here would be the fasciculus gracialis. Okay, so the pons. It's going to be the ventral part of the metencephalon. It's going to contain the motor nuclei of cranials 5, 6, and 7, along with the sensory nuclei of cranial nerves 5, 7, and 8, along with the pontine nuclei. So the axons of the pontine nuclei will grow transversely and form the transverse pontine fibers along with the middle cerebellar peduncle. Right here. And then the fibers will go straight up to the cerebellum or straight across to the cerebellum, and they connect and make the middle cerebellar peduncle. The cerebellum is going to be the dorsolateral part of the alar lamina of the mesencephalon, and it extends medially and dorsally to form something called the rhombic lips. They fuse over the 4th ventricle and dorsally to make the cerebellum. And by the end of the 4th month, the fissures and the folia form. So there's the fissures, and then inside where it branches off, that's the folia. Neocrest cells from the matrix will migrate to the surface to form the cortex. and then other neuroblasts will come in and follow. And fibers from the olive, the spinal cord, pons, vestibular nerve will make the inferior peduncle. And then in the upper midbrain, the superior middle peduncle tectum, and it's going to be the sensory nuclei only. While in the lower midbrain, the inferior peduncle tegmentum is going to be just sensory, or is going to be both sensory and motor. So upper sensory only, lower sensory and motor. Midbrain is going to form the cerebral aqueduct, and the alar lamina will migrate to the back and invade the roof place to make the colliculi. So it's going to go over here, make the colliculi. The basal lamina, or substantia nigra, and tegmentum, the marginal layer of each basal lamina will make the crus cerebri. And the basal plate, once again, is going to be motor, and alar is going to be sensory. And it also, the substantia nigra is just the special one because it has both sensory and motor fibers. Forebrain is going to be made out of like the prosencephalon, and that makes the diencephalon. And the cavity is going to be the third ventricle. The lateral walls are the hypothalamus sulcus, which is the continuation of the sulcus limitans, and that will make the ventral and dorsal regions. Ventral region is going to be the hypothalamus, and the dorsal region is going to be the thalamus. And that will create the medial and lateral geniculate bodies. And it's important to keep in mind, all of this is all happening at the same time. So while the forebrain is being formed, the tegmentum is being formed, the mammillary bodies, all those are all being formed. Okay, so the diencephalon is where the neurohypophysis is, or the pineal gland, and the pineal gland. The adenohypophysis will grow up from the Rathke's pouch. So that comes from the mesoderm. This comes from the neurocrestals, and they kind of meet. Like this grows up, this goes down. They meet when the third ventricle enlarges and pushes the diencephalon down, and then eventually it will make the pituitary gland. The epithalamus will make the pineal gland and the habenular nucleus, and then the pineal gland will grow from the roof plate. And the hypothalamus is going to be made of the neuroblasts in the lower parts of the alar plate that has been pushed down to the floor. The telencephalon is made out of the lateral terminalis, which is going to be the closed anterior neuropore that happens on day 25, and that lies between the cerebellar vesicles. And the cavities of the hemisphere communicate with the diencephalon through the intraventricular foramina of Monroe. The cerebral hemisphere grows forward and then up and then back. And then as it pushes backwards, the choroid plexus is going to protrude into the choroid fissure to the lateral ventricle. And then the hippocampus is going to be connected to the hypothalamus via something called a fornix. Hippocampus is going to be pushed downwards when the neocortex enlarges. And then in the 12th week of development, pyramidal granular cells form. So the limbic system is the continuous growth of the cerebral hemisphere, and that will make the lobes. The corpus striatum slows, and the insula will form. And that occurs in the first part of the fetal life. The cerebral hemisphere grows, the gyri and the fissure and the sulci form. And then in the 12th week, the cortex becomes very cellular. And at the term, the neuroblasts have differentiated and they are stratified now. And then the motor cortex will have the pyramidal cells, while the sensory will have the granular cells. The commissure is going to bridge the two hemispheres. The anterior commissure is going to be the first to form, and that connects the olfactory lobe and the temporal lobe from one side to the other, so on both sides of the brain. The fornix will connect the hippocampi to each other, and the corpus callosum is going to be the last to develop. It connects the frontal along with the parietal lobes, and it arches over the third ventricle. A septum pellucidum is going to migrate backwards, and then the optic chiasm forms, and all of this develops very quickly. This develops within days. So the corpus striatum is going to be a sensory motor complex, and it develops in the floor of the telencephalon. And then when the neocortex develops, a tract has to pass through it and divide it into the caudate nucleus, which is going to be your dorsomedial area, and the lentiform, which is going to be your ventral lateral area. The lentiform nucleus will divide into the globus pallidus and the putamen. So here is your putamen, here's your caudate nucleus, and here is your, the very center is going to be the globus pallidus. Okay, so the peripheral nervous system made from the neurotube, neurocrystals, and ectodermal placodes, and it forms from like head to foot. It's bipolar only at the spinal ganglion of the cochlear and the vestibular ganglion of cranial nerve 8. The rest are going to be myelinated unipolar cells. And Schwann cells are going to come from the trunk and caudal area. The sympathetics of the peripheral nervous system arises from the thoracolumbar region. And the parasympathetics will arise from the craniosacral region. So this would come from like the entire cord. Myelination is done due to the oligodendrocytes and the Schwann cells. And in the 4th month, the spinal cord will start to myelinate. The first to myelinate is the sensory fibers, and then the descending motor fibers will be the last ones to form. 6 months after birth, it starts in the brain, and it begins only in the basal ganglia, and it finishes by the 2nd year. Okay, so the neurotube defects usually occurs during the 3rd week, and due to folate deficiency, and that could be either spina bifida, anencephaly, and encephalocele. Encephalocele is going to be the cyst, right? The Arnold-Chiari malformation is when the medulla and cerebellum is going to herniate into the foramen magnum. Hydromelia is going to be the enlargement of the spinal cord. Syringocele is going to be the enlargement of the central canal. And syringomelia is going to be abnormal cavities near the central canals. The Dandy-Walker malformation is the blockage of the median and lateral apertures of the 4th ventricles, and it causes the enlargement of the posterior cranial fossa, and it's going to compress the cerebellum. It's not going to allow it to form. Holoprosencephaly is going to be the failure to separate the forebrain vesicles, and it's usually in weeks 5 to 6 when the forebrain is forming. It's really common in trisomy 13 and fetal alcohol syndrome. And of course, it depends on the penetrance. Sometimes it can be mild, and sometimes it can be incompatible with birth. Or incompatible with life. Lissencephaly is going to be the failure of neuronal migration, and it results in a smooth brain. No chiari will form. This is generally incompatible with life. Alright, to go through the chiari malformation some more, there's 4 different types. The first type is going to be when the inferior part of the cerebellum herniates through the foramen magnum, and it's only a little bit. Sometimes you can hardly see anything. It's just like a tongue-like projection. Type 2 is going to be the Arnold-Chiari malformation. This is much more serious. Type 3 is very severe. There's herniation of the cerebellum and brainstem into the vertebral canal, so not just the cerebellum and the medulla. Type 4 is when the cerebellum is absent or underdeveloped, and that is incompatible with life. All of these can result in various forms of hydrocephalus. So this one, though asymptomatic, usually any kind of trauma can really cause it to be present. Okay, so that's it for now. It's a long video, but thank you for your attention and good luck studying.